Bismillah ar-Rahman Rahim, 9700 A-Level Biology, paper 2-2. This is the second video in which we will solve the rest of the questions which were not done in video 1. Now, starting with question number 3, lysozyme is an antibacterial enzyme that was discovered in 1921 by Alexander Fleming, the scientist who discovered penicillin. Lysozyme catalyzes the hydrolysis of glycosidic bonds present in peptidoglycan molecule to form smaller products, NAG and NAM, N-acetylmuramic acid and N-acetylglucosamine. Part A says, before the induced fit hypothesis was proposed in 1958, scientists believed that the lock and key hypothesis explained how lysozyme catalyzes the hydrolysis of peptidoglycan to its products. Draw labeled and annotated diagrams in the space provided to show how the lock and key hypothesis were used to explain the mechanism of action of lysozyme on peptidoglycan. Now, what are the 3 marks? How are you going to get the 3 marks? The 3 marks were for number 1, active site with a specific shape as part of the molecule drawn and active site labeled or included. So I didn't include it in an annotation. So active site, you would have to label that. So let's label that active site. Let me give it another color. So this would be the active site. Complementary shape peptidoglycan drawn and peptidoglycan labeled. Enzyme substrate complex drawn and labeled. Lysozyme with same shaped active site and 2 products, NAM and NAG drawn. So any 3 of these and you got your 3 out of 3. Part B of the question says lysozyme and penicillin can be described as antibacterial agents. Compare lysozyme and penicillin to show the similarities and differences between these 2 antibacterial agents. Both of them cause cell bursting or cell lysis, and the reason why being it weakens or destroys the cell wall of the bacteria. Now, difference is penicillin is an antibiotic, is an enzyme inhibitor. Penicillin inhibits transpeptidases. Penicillin prevents formation of the peptide cross bridges. Penicillin only effective when cell is growing and synthesizing cell walls, but lysozyme is effective at all stages. Lysozyme is a much larger molecule as compared to penicillin. So these would be the 2 similarities. Both cause cell bursting and both weaken and destroy the cell wall. Then we come on to the differences. Now the differences is lysozyme is a protein. It's an enzyme. So penicillin is an antibiotic. So we're not writing that in every situation. It's compare lysozyme and penicillin. So you've got to tell me what you're talking about. So penicillin is an antibiotic. Lysozyme is a protein. You could have written that. Penicillin inhibits transpeptidase, an enzyme. And penicillin prevents the formation of peptide bridges. Penicillin is only effective when the bacteria is synthesizing cell walls, that is when the bacteria is growing. But lysozyme is effective at all stages. So only effective when bacteria is synthesizing cell walls, but lysozyme is effective at all stages. Then we come to question number 4. In the mesophyll tissue of leaves, products of photosynthesis can be used to synthesize organic compounds such as polysaccharides, cellulose, and some amino acids. A source of nitrogen for amino acid synthesis can be provided by nitrate ions that have been taken up in the roots and transported to the leaves. Describe the structure of a cellulose molecule. Very simple, direct question. Monomers of beta glucose, 1,4 glycosidic bonds, each beta glucose rotated by 180 degree compared to the adjacent beta glucose. It's linear, or you can say straight chain molecule, and it is unbranched. So straight, very simple points which everybody must know for cellulose. So monomers of beta glucose, 1,4 glycosidic bonds, each beta glucose rotated by 180 degree compared to adjacent beta glucose, linear straight chain, unbranched. Part B of the question, studies of nitrate uptake and nitrate metabolism help to provide information to scientists who are investigating ways to increase the yield of crop plants. First step of nitrate metabolism in leaf cells is the reduction of nitrate to nitrite catalyzed by the enzyme nitrate reductase. The activity of the enzyme can be studied by detecting the presence of nitrite formed. Researchers have found that adding nitrate to leaf tissue results in an increase in messenger RNA molecules of the gene NR, which codes for nitrate reductase. State one benefit to leaf cells of an increase in mRNA molecules of gene NR after the addition of nitrite. So it is going to be an increased rate of translation of the enzyme, increase in nitrate reductase synthesis or the concentration. Now enzyme only produced when needed. So more nitrate is reduced to nitrite. So more nitrite is formed, leading to increased supply of nitrite for amino acid synthesis. So the benefit or to leaf cells of an increased mRNA of gene NH, you have to give me all those or one of all those points. So increased rate of translation of NR increases enzyme concentration, so more nitrite formed for amino acid synthesis. One method used to detect the presence of nitrite formed from the reduction of nitrate in leaf tissue involves using an inhibitor to prevent nitrite from taking part in further reactions in the leaf. So nitrite will keep on accumulating. Immersing the leaf tissue in a solution containing a colorless test reagent. The nitrite from the leaf tissue, 10 nitrites or 100 nitrites. Now give yourself an example to yourself. The nitrite from the leaf tissue enters the surrounding solution, changing the color of the solution to magenta. So from colorless, it will become magenta, red, purple. Suggest by using a colorimeter can improve the method to detect the presence of nitrite. Colorimeter gives you a reading on the intensity of the red, purple, magenta color. So this will improve the accuracy of the results. You're not just going to look at by eye, but you're going to get a value. The results are not subjective. Now this will provide a quantitative result, means it will provide a numerical value. Say the absorption is 10 or 20 or 30, so the darker the color, the more the absorption will take place. Different intensities or shades of color of the red purple relate to different concentrations of nitrites, and then we can produce a calibration curve, and also we can detect very low concentrations with the help of a colorimeter. So provides numerical value, different shades relate to different concentrations, can produce calibration curve, can detect very low concentrations. Part C, in the leaf, transport of amino acid from mesophyll cells to companion cells involves using a number of different membrane transport proteins called amino acid transporters. There is evidence that amino acids can move from the apoplast into the cytoplasm of a companion cell using the same transport mechanism that is used in the sequence of events that occur which allow amino acids to be transported from the apoplast into the cytoplasm of a companion cell. But because they've said it's the same as sucrose, so then this is a direct question, and this has come, I think, a million times in all the past papers, and the mark schemes are so much the same. Protons moved by active transport. Protons moved into the cell wall, then proton gradient build up in the cell wall, then protons move back by facilitated diffusion from a higher to a lower concentration, and protons co-transport amino acids. Or you can say amino acids move with the protons through a co-transporter protein. So amino acids are transported against their concentration gradient. But please remember, it is co-transport which is actually taking place. So the points are very basic and easy. Protons moved by active transport, protons moved into cell wall, proton gradient builds up in cell wall, protons move back by facilitated diffusion, protons co-transport amino acids transported against the concentration gradient. Then a very interesting question, part 2, suggests why amino acid transporters are not needed to move amino acids from the companion cell into a phloem sieve tube element. Why? Because the plasmodesmata present. The plasmodesmata present between the companion cell and the phloem sieve tube element. So plasmodesmata present. That is what a cytoplasmic thread between between the two cells, the companion cell and the phloem sieve tube element. Nitrate ions, the root tissue can be analyzed to determine the quantity of nitrate taken up over a set time period. D part of the question says excised roots of the crop plant maize, Z maize, were placed in 3 different concentrations of nitrate solution, 0.2 millimole dm cube, 1.0 and 5.0. So it's a 0.2 and a 1.0 and a 5 mole dm cube. The solutions were maintained at 30 degrees Celsius and was aerated, air pumped in, to provide a continuous supply of oxygen to the root tissues. Night for 5 hours. The results are shown in figure 4.1. Now let's look at the results. We have time in hours, we have to 5 hours, and nitrate uptake in micromole per gram fresh mass. So you can see how on 1, then 2, and this is for 0.2. And so we have this for 0.2. Let's look at this for 0.2. Let's look right at the end. How much is it? Okay, so it's gone up to nearly something like 6 point something. And the 1 mole has gone up. Wow, it's gone up a lot. And it's gone up to something like between a little less than 18. And then of course we have the 5 mole, which has gone up and has gone up. And then you look at the gradients and you look at the last at 5 hours, what was the final. So you need to be writing all those down. So this is 20, and then of course we write down the others as well. Figure 4.1 shows that the rate of nitrate uptake is very low initially and then increases for all 3 concentration of nitrate solution tested. Describe the differences in the rates of nitrate uptake for 5 mole millimole dm cube nitrate solution compared with 0.2 nitrate solution between 2 hours and 5 hours. Now they've got to, and this is for 2 marks. So we only have to compare 0.2 and 5 and between 2 hour and 5 hour. So we are only comparing 0.2 and 5 mole, right? And we are comparing it, interesting is between 2 hours and 5 hours. So we're comparing it this area, 2 hour and 5 hour. So look at the difference between the blue line and the red line. So this is a very, very sharp increase between 2 and 3. This is a gradual increase between 2 and 3. And then the final year was 6.4 and the final was 20 micromole per gram. You see where the main thing which was the difference was the rates of nitrate uptake. So if you look at the graph, 0.2 mole is a constant rate. 0.2 mole, there is a constant rate. This one is a constant rate. And versus the 5 mole rate is not constant. You see the rate keeps on changing. You see there's a rate here, it's like this. Here there's this sharp increase and then is a gradual increase. So rate is not constant. So 0.2 constant rate versus 5 millimoles dm cube rate not constant. Then 0.2 is a lower rate than 5. So lower rate than 5, or you could have said 5 millimoles is a higher rate. And then you could do a calculated rate to support these answers. You could calculate the rate. So 0.2 constant, 0.5, sorry, 5.0 rate not constant, 0.2 lower rate than 5.0 millimole. And you could also calculate, that's why I've left this in brackets, is that you would calculate the rate per on the 2 to 5 means in 3 hours you would calculate the rate at the beginning and then at the end and then divide it by 3. Then part E of the question, the nitrate uptake of excised maize roots and was investigated under different conditions. Table 4.1 shows details and results for a control experiment and 4 modified experiments, 1, 2, 3 and 4. The same concentration of nitrate solution was used throughout for all the experiments. All the results were taken after a set time period. So experiment 1, 30 degrees, aeration, no nitrogen gas bubbled through instead of oxygen. Nitrate uptake was 0.4 micromoles per gram. In experiment number 2, the temperature was 3. Aeration was yes. Additional substance present in nitrate solution, none. And it was 0.6. In 3, it was 30, yes. Protein synthesis inhibitor, 1.4. 430, yes. Antibacterial compound, 10. Control was 30 temperature, yes. Aeration was present. No additional substrate in the nitrate solution, and it was 10.9. The results for experiments 1, 3, 3, and 4 and table 4.1 can be compared to the results for the control experiment. So we have to compare this with the control. Now it says describe how comparing each of the results with the control provides information about how nitrate ions are taken up by the root cells and the factors affecting the uptake of nitrate ions. So how do we compare the results and what do we get? We're getting is that there's active transport, and the comparison of the experiments and control can be a whole lot of things. Oxygen required for uptake, no uptake or almost no uptake in low temperatures, then protein or enzymes are required for uptake, then slightly lower, very similar uptake without any bacteria, or very similar uptake with bacteria control. And you could have given me a comparative result to support one of the marking points too, which is no uptake in low temperature. So you know that at 3 degrees Celsius here, it was 0.6. So marking point 2 is almost no uptake in low temperature at 30 degrees, or you could have said uptake higher at 30 degrees in control. And control, the uptake was 30 and it was 10.9, and here it was 0.6 at very low temperature. So active transport, oxygen required for uptake, almost no uptake at 3 degrees Celsius, proteins required for uptake, very similar uptake with bacterial control. And then if you give me the figures for 3 degrees and 30 degrees Celsius. In this question, you could also have given more discussion points as like aerobic respiration needs oxygen, lower temperature means lower activity of enzymes involved in respiration, and then ATP needed for active transport, some facilitated diffusion because some uptake without oxygen, then protein synthesis needed for transport proteins, then lower temperature will reduce the rate of facilitated diffusion, bacteria will have little or no effect on uptake. So there were other lot of discussion points which I have not written, which you could have given me as well for this part of the question. Please remember that. Going to question number 5. During interphase of the cell cycle, individual chromosomes cannot be seen within the nucleus. The genetic material is termed chromatin during this stage. Changes occur to the chromatin during mitosis so that chromosomes become visible. State what happens to the chromatin so that individual chromosomes can be seen during mitosis, especially prophase of mitosis. So what is going to happen is condensation, or you can say becomes more compact. You can even say coiling or supercoiling. I like the word becomes shorter and fatter. Oh, so all these words you could have used for explaining why now they're visible and previously why they were not visible. So condensation becomes more compact. Figure 5.1 is a transmission electron micrograph of the two human chromosomes at metaphase of mitosis. Describe the structure of chromosomes at metaphase, such as the two chromosomes shown in figure 5.1. Each has two sister chromatids. Chromatids are joined by a centromere. Each chromatid has a single DNA molecule. DNA is associated with histone proteins and telomeres at the ends of the chromatids. Each has two sister chromatids joined by a centromere. Each chromatid has a single DNA molecule. DNA associated with histone proteins, telomeres at ends of chromatids, chromosomes highly condensed. Question number 6. Some people who are infected with HIV have HIV/AIDS. State the full term for HIV, human immunodeficiency virus. It's all underlined, so either you got this right or you just didn't get it wrong. Please try to get the spellings correct. Part B of the question, following transmission of HIV, early diagnosis of infection and treatment with antiretroviral therapy helps to control the spread of the pathogen and prevent HIV/AIDS. Suggests why treating people who have developed AIDS with ART may help to reduce the number of overall deaths from infectious diseases such as cholera, TB, and malaria. Well, basically, what does it do? Helps to increase the number of T lymphocytes. So this maintains or improves the immune response or the immune system. So this increases chances of recovery from infectious diseases or opportunistic diseases. So decreased risk of having other opportunistic diseases, and this will decrease the viral load, so it will reduce the number of HIV. Helps to increase the number of T lymphocytes, so improves the strength of the immune response, increases chances of recovery from infectious diseases, decreased risk of other opportunistic diseases. Studies suggest that people who are infected with HIV may be at a higher risk of heart disease. One cause coronary artery bypass graft is a surgical operation that uses healthy blood vessels to divert blood around diseased sections of coronary arteries. The main choice of blood vessel to use for a bypass graft is known as the internal thoracic artery. Figure 6.1 is a diagram of external view of the heart to show a double bypass graft. The blood vessels used in the bypass graft shown in figure 6.1 are the great saphenous vein from the leg and the internal thoracic artery. You can see the bypass here. Now, what is the question they want you to? Draw across an X on figure 6.1 to show an area of the right coronary artery that has been bypassed. So this is the area which has been bypassed. So all this area would be the damaged area. And then you can see this is the bypass. So this is the right side of the heart. So the question was, draw across to show an area of the right coronary artery that has been bypassed. So then that would be this area. So you see this has been bypassed. So this would be the area which is affected. That's why this area has been bypassed. This is the coronary artery which has been bypassed. And now we come to the last part of the last question. After surgery, the wall of the great saphenous vein becomes thicker. It's a vein taken from some part of the body. Suggest and explain why it is important for the wall of the vein to become thicker after surgery. Well, it can withstand the higher blood pressure, so it's less likely to burst. The blood vessel may last longer. Then the tunica media, there'll be more smooth muscle or thicker smooth muscle and more elastic fibers. So of course, this all will help on the onward movement or the faster flow of blood. So this will result in a thicker tunica externa or the tunica adventitia, which is also called. So can withstand high blood pressure. So blood vessel may last longer, more smooth muscle or more elastic fibers, helps faster flow of blood and a thicker tunica externa. So it says suggest and explain why it's important for the wall of the vein to become thicker. Well, it's become thicker. Why should it become thicker? The wall of the veins are usually not thicker. But then why they're developing? So what is making it thick? More muscle in the wall, tunica media, tunica externa, or the tunica adventitia.